Thanks, John, for the invitation to come and talk to you today. So I'll talk about what is my real interest really is which is cholesterol metabolism, and really the importance of oxysterols in biology. So as John said, next slide. Oh, I should be before I go on, I should give a disclosure that myself and my colleague are listed as inventors on this patent, kitten method quantitative detection of steroids, and we're shareholders in this company, Calisthenics. So as John mentioned, we're in Wales in the UK, so we're in South Wales. So this is Swansea, just by here, this in this bay, and the university's bang on the coast. And if it were a nice day and you went to the entrance of the university and looked right, this would be your view. So it really is it's an excellent place to live, and an excellent place for students. So it's a really lovely location. Okay, to the talk. So sort of overview of the talk. We'll start with cholesterol, which is the archetypical sterol. We'll look a bit about the biosynthesis and metabolism, and then we'll talk about the molecules that I'm really interested, which are the oxysterols, and then we'll talk about their analysis by LCMS. And then we'll look at some sort of applications of why oxysterols can actually be important. So we'll look at a neurodegenerative disease. We'll look at Huntington's disease, and then we'll look at what I'm doing a lot at the moment, which is inborn errors of cholesterol metabolism, and looking at trying to improve the diagnosis. And at the end, we'll have some mass spectrometry imaging of cholesterol and oxysterols. Okay. So a few words about cholesterol itself. So here's the molecule. So it's abundant in all animal cells. 10 femtomoles per cell. That's about 6 times 10 to the 9 molecules. So it's an abundant molecule, and most of the cholesterol in a cell is actually in the plasma membrane, where it can make up about 45 mole%. So it really is. It's abundant, and it's mostly in that plasma membrane. And then cholesterol metabolism, it can lead to steroid hormones and the bile acids. What's really important, I guess, or significant about cholesterol is that its levels in a cell are really tightly regulated, and they're regulated by a mechanism called convergent inhibition, which was worked out by Nobel Prize winners Goldstein and Brown in Dallas a number of years ago. So I put key references at the bottom here, so you'll see Brown and Goldstein mentioned many times. So if we look at sort of cholesterol synthesis, we start with acetyl-CoA and we end up with cholesterol, and we've got about 20 enzymes involved in the pathway, and these enzymes are really regulated by the master transcription factor SREBP2, or sterol regulatory element binding protein 2. And each of the genes of these enzymes in the cholesterol biosynthesis pathway has got a sterol regulatory element, and SREBP2 binds the sterol regulatory element and activates target gene transcription. The LDL receptor also has a sterol regulatory element, and SREBP2 will also regulate the transcription of the LDL receptor. So this SREBP2 is a real key regulator of cholesterol biosynthesis. So Goldstein and Brown actually worked out how SREBP2 works and how it regulates cholesterol biosynthesis. So SREBP2 sits in the endoplasmic reticulum, and it sits next to a protein called SCAP, which is a transport protein. And the function of SCAP is to transport SREBP2 from the ER to the Golgi. Once SREBP2 gets in the Golgi, it gets processed by 2 enzymes, S1P and S2P, to its active form of the transcription factor. That then translocates to the through the nucleus and activates target gene transcription. Now, once a cell has got enough cholesterol, or even excess cholesterol, then cholesterol in the ER will bind to SCAP. And when it binds to SCAP, SCAP binds to INSIG. Now, INSIG is an anchor protein. So INSIG always sits in ER. So INSIG then sort of anchors SCAP and ultimately SREBP2 in the ER. So it can't be transported to the Golgi, so it can't get processed into the active transcription factor, so cholesterol biosynthesis is turned off. So if we elevated cholesterol levels, cholesterol synthesis turns off. When the cholesterol levels drop down, then cholesterol synthesis goes back on. So this is convergent inhibition. It was all worked out, as I said, by Brown and Goldstein. The molecules that I'm interested in, these oxysterols, they can also regulate cholesterol synthesis. It's particularly the side chain oxysterols, what we'll look at in a minute. And they sort of fine-tune cholesterol synthesis. So they work in a slightly different way in that the oxysterols, so for example, 25-hydroxycholesterol, instead of binding to SCAP, they bind to INSIG. And when they bind to INSIG, INSIG binds to SCAP, gets held in the ER, and again, we turn off cholesterol synthesis. So these oxysterols are probably fine-tuners. Okay, let's now talk to you about these oxysterol molecules, the ones that I'm really interested in. So the side chain first of all, an oxysterols are oxidized forms of cholesterol. So essentially, almost every one of the carbons in cholesterol can be oxidized to give an oxysterol. And the side chain oxysterols is when we've got oxidation in the side chain. So there are a lot of oxysterols, and if you just take a look in plasma, and you might look at what we call the primary oxysterols. So these are the oxysterols with just 1 degree of oxidation. You see a lot of them. So for example, this one, 22R hydroxycholesterol. This is made in the steroidogenic organs, and it's the first step of steroid hormone biosynthesis. This one, 24S hydroxycholesterol, so we've moved the hydroxy from 22 to 24. This one's only made in brain. Actually, it's only made by neurons, and it's exported from the brain into the bloodstream. 25-hydroxycholesterol, this is normally a minor oxysterol, but if you have an infection, then macrophages make a lot of it. So this is only observed when you have an infection. This is 26, also called 27-hydroxycholesterol. This is made by just about all cells, and it's formed in the first step of the acidic pathway of bile acid biosynthesis. 24-25-epoxycholesterol, this can be formed in parallel to cholesterol, and it's involved in neurogenesis. 5,6-epoxycholesterol, this seems to be linked to cancer. Then we've got 7-alpha-hydroxycholesterol. This is the first step in what's called the neutral pathway of bile acid biosynthesis. And at the top, we've got 7-beta-hydroxycholesterol and 7-oxocholesterol. These are precursors of hedgehog signaling molecules. So you can see there are a lot of these molecules, a lot of isomers, and many of them are, in fact, all these I show you here are actually biologically active. So these are what I would call primary oxysterols, where you've got one stage of oxidation, but then you've got the secondary oxysterols, where you can have a further step of oxidation. So if we just take 24-hydroxycholesterol as our example, so 24-hydroxycholesterol can be metabolized further to 7-alpha-24-hydroxycholesterol onto 7-alpha-24-26 trihydroxycholesterol, and so on. So the number of potential oxysterols, you can see it's getting huge, and things are even more complicated as some of the cholesterol precursors can also make oxysterols. So if you look at the Candice-Russell pathway, 7-dehydrocholesterol is a precursor to oxysterols. If you look at the block pathway, desmosterol is a precursor to oxysterol. So this oxysterolome is absolutely massive. So why why is anyone interested in oxysterols other than them being oxidized forms of cholesterol and its precursors, and they're formed always in the first steps of cholesterol metabolism. They're also intermediates in the bile acid steroid hormone biosynthesis pathway. So why are they interesting? First of all, because they've got this added oxygen functionality, they can cross membranes much more easily than cholesterol. Many of them, particularly the side chain oxysterols, they're biologically active, so they interact with receptors. And I think almost certainly they have important paracrine, autocrine, and even perhaps endocrine signaling properties. So I regard them as signaling molecules. So I think probably the most important function, at least of the side chain oxysterols, is in the regulation, this fine tuning of cholesterol synthesis. So for example, this 25-hydroxycholesterol, this can be made by a cell, secreted by a cell, and have an effect on a neighboring cell. Because 25-hydroxycholesterol and these other oxysterols, they'll cross membranes really quickly. So it can shoot across a plasma membrane, get to the ER, and turn off cholesterol synthesis in a neighboring cell. It'll also activate SOAT. So SOAT is the enzyme that converts cholesterol into cholesterol esters. So this 25-hydroxycholesterol, it can turn off cholesterol synthesis in the neighboring cell, and it also encourage cholesterol to be converted to cholesterol esters. So it'll reduce the cholesterol content of a cell. So this might be a neighboring cell, or if the oxysterol is made in the cell itself, it'll be a sort of a cell endogenous mechanism. What's more, these side chain oxysterols, they'll also interact with nuclear receptors, particularly the LXR nuclear receptor or liver X receptor. So the side chain oxysterols, here illustrated by 25-hydroxycholesterol, that will bind to the liver X receptor, activate target gene transcription. One of the target genes is ABCA1, so we get ABCA1 production. Now, ABCA1 is a cholesterol transport protein. So ABCA1 will pump cholesterol out of the cell. So the effect of the oxysterol on the cell through LXR, again, is reducing that cholesterol level in the cell. So oxysterols almost certainly are really important in this fine-tuning of cholesterol levels in cells. Okay, so that's a little bit about why they're interesting molecules and perhaps why they're important. So let's now move on to, I guess, analysis, which is our expertise. So here's a typical oxysterol. This is 26-hydroxycholesterol, also called 27-hydroxycholesterol. And it's not so easy to analyze by mass spec, because in mass spec you need to ionize the molecule. And this type of molecule, it's neutral, so it's not so easy to ionize, particularly if you're using modern methods of LCMS. So we and a number of other groups have thought of ways to actually improve the ionization of this type of molecule. And we came up over 20 years ago with a method to improve the ionization, which is based on the Girard hydrazine reagent. So I'll just walk you through our little derivatization method. So this works on oxysterols, also works on cholesterol precursors. So we call the method enzyme-assisted derivatization for sterol analysis, or EDSA. So you'll see why it's got the name in a minute. So this is our typical oxysterol, and most oxysterols have got this 3-beta-hydroxy delta-5 group. So what we do, we take our sterol extract and we treat with bacterial cholesterol oxidase. So you can buy this from Sigma. So cholesterol oxidase will convert the 3-beta-hydroxy delta-5 to a 3-oxo delta-4. So this is the actually classical Richmond chemistry used in the measurements of actually cholesterol in the bloodstream. So once we've got the 3-oxo delta-4 group, we can now react it with our Girard hydrazine reagents. The Girard hydrazine reagents it's a hydrazine, and it's got this pyridine group, and it's got quaternary nitrogen, which has got a positive charge. So we like to use this deuterated Girard reagent. So the hydrazine will react with the ketone to give a hydrazone, and then we tag on this positive charge onto our molecule. So now we've actually made it charged. So that means the mass spec will love it, and you get a lovely big peak. So here's a little cartoon of what goes on. So without derivatization, you get a peak, but it's not a very good peak. We treat with cholesterol oxidase that introduces the 3-oxo-delta-4 group. We then add our Girard reagent. Now, whenever we use the enzyme, we use the deuterated Girard reagent, the D5. So that clicks onto the ketone function. We get hydrazone, and the signal goes up by a factor of 100 to 1,000, depending on what mass spec interface you're using. So this derivatization, it has two advantages. One is the signal, and also the specificity, because we're using that enzyme reaction at the front, so it's adding specificity to the derivatization. So this is really useful. And what's more is when we try to fragment the molecules using MSMS or MS to the 3, you get really nice fragmentation patterns. So here's our molecule that's been ionized. If we then do first step of fragmentation, we lose the pyridine group. Then this is unstable and will fragment further. And when it fragments further, we get fragmentation in the ring system, and we also get fragmentation in the side chain. So that means you get fragmentation spectra, which are diagnostic for the parent molecule. Okay, let me sort of illustrate this now with if we look at the monohydroxycholesterols in plasma. This is typical plasma chromatogram we get looking at monohydroxycholesterols. So the first peak is 24 hydroxycholesterol, second peak 25 hydroxycholesterol, third peak is 24 hydroxycholesterol. This is a slight disadvantage of the method, is that we quite often get 2 peaks for a given molecule. This is because we can get cis and anti conformers about this new hydrazone bond that we've introduced. So the hydrazone bond, the derivatization could be here, or alternatively it could be up here, and we're able actually to resolve the two conformers. So then we've got 26, and that also gives 2 peaks, and 7 alpha, that also gives 2 peaks. So this is advantageous in that you've always got 2 signatures, but it's a disadvantage in you do complicate the chromatogram. So if you look under one of the peaks, you get a nice spectrum, a nice plain mass spectrum for the molecular ion. So if we then go and look at the fragmentation spectra, first look at the fragmentation of 24 hydroxycholesterol. So at the bottom end of the spectrum, you get fragment ions which come from ring fragmentation. So we've got fragmentation in the A and B rings. And in the middle part of the spectrum, you get fragment ions which come from cleavage of the side chain. And the top part of the spectrum, you get neutral losses of bits of the derivatization group and water. So this is 24 hydroxycholesterol. If you look at 26 hydroxycholesterol, the fragmentation spectrum is similar, but it's different in that the pattern at the top end of the spectra is different. So here, 437 is bigger than 427. In 26 hydroxycholesterol, you've got the other way around. If we look at 25 hydroxycholesterol, if we look at 25 hydroxycholesterol, that's dominated by just the one fragment ion. Everything else is small. And if we put the hydroxy in the ring system, we get a completely different fragmentation pattern down at the bottom end of the spectrum altogether. So this is useful. It really, when we look at data, we can really be sure what each peak is. And if we get a new peak, we can make a pretty good stab at identifying what it is as well. So these are the advantages of the derivatization method that we use. I guess there are always complications with derivatization. And some molecules, actually, some oxysterols naturally possess a 3-oxo 4-ene group. So if we're interested in these that naturally possess a 3-oxo-4-ene group, then we don't need to bother with the enzyme at the beginning. So we can skip the cholesterol oxidase. And when we're interested in these molecules, the Girard reagent that we use is the D0 Girard reagent. None of those deuteriums are. And at the end of the pathway, at the end of the oxysterol metabolism pathway, we end up with bile acids. Now, bile acids are actually transparent to our derivatization because our derivatization requires a ketone or a 3-beta hydroxy, and these have got 3-alpha hydroxy. So the cholesterol oxidase won't work on these. But bile acids themselves, they've got a natural carboxylic acid group, which gives a good ion in the negative ion mode. So what we do, if we're interested in the bile acids, we just flip the mass spectrometer into the negative ion mode, and we can detect them. And you know, with modern mass spectrometers, you can have the one run and just go flip between positive and negative ion mode in the one run. So we can get the bile acids when we're interested, you know, get them for free by flipping into negative ion mode. Okay, so this is just our overview of our protocol. So we work with tissues, mostly brain or liver, and fluids, mostly CSF and plasma. So we do an extraction of the sterols into ethanol. Then we start with a little column, a little C18 column, where we separate the oxysterols from cholesterol, because cholesterol is always really abundant. There's usually 1,000 times more cholesterol than the oxysterols. So we like to get rid of it, get rid of that cholesterol straight away. So we then take our oxysterols and split them into two fractions, an A fraction and a B fraction. The A fraction, we add the cholesterol oxidase and the deuterated Girard reagents. This will be applicable for those oxysterols with a 3-beta hydroxy group. To the B fraction, we skip the enzyme, so this, and add the non-deuterated Girard reagent. So this will only work now on those oxysterols that have got a natural 3-ketone group. And then we actually do a second little cleanup step, just to remove excess derivatization reagent. We can then add the A and the B fractions together and do our LCMS. So if a peak is derivatized with the D5, it'll always obviously be 5 mass units than higher than a peak derivatized with the D0. So it's quite easy to differentiate which peaks come from the A fraction, which peaks come from the B fraction. We have actually made a lot of different isotope labeled versions of the Girard reagent. So we could actually do a 6-plex experiment if we wanted to. But in reality, we only ever do a duplex using the D0 and the D5, because while we can make these more exotic isotope labeled Girard reagents, you know, basically it's too expensive to bother. So we only ever do a D0 and a D5 together. Okay, so that's our our methodology, and I've introduced you to oxysterols. So now let's look at how oxysterols and cholesterol, you know, might be interesting or are interested in the real world. So we'll pick on Huntington's disease. So this is it's a rare autosomal dominant neurodegenerative disease, and it results from actually I think it's a CAG, not a GAG. A CAG, it could be a GAG. I'm not quite sure. Anyway, it results in I think it's CAG repeat in the HTT gene. So this results in a polyQ in the HTT protein. So this genetic modification it results in Huntington's disease. So Huntington's disease, it's a horrible disease. So first of all, it's autosomal dominant. So if one of your parents has got it, you know, there's a 50% chance that you will get it. Onset is often in middle age, and it's a movement disorder, and also it results in cognitive decline. There's no cure, and you know, death of Huntington's disease is the result. So why might we be interested in Huntington's disease? Well, it's thought that Huntington's disease is linked in some way to disturbed cholesterol homeostasis in the brain. And how it's disturbed is somewhat controversial, and the data is contradictory. And the sort of two groups of thought now have developed of how changing cholesterol metabolism or changing cholesterol levels in Huntington's disease patients could actually result in a cure. So there's one group of thought which comes from Milan in Italy, and they believe that actually increasing the cholesterol levels in the striatum of the brain could actually be a cure for Huntington's disease. So they've got rather quite convincing mouse data for this. And then on the other hand, there's a group in Paris in France who think another way, and they think that actually instead of increasing cholesterol levels, it might be better to increase cholesterol metabolism. And they've been focusing on ways to increase cholesterol metabolism as a way of treating Huntington's disease. And actually, they've got a clinical trial running in Paris at the moment where they're actually using AAVV to put an enzyme into the brain to increase cholesterol metabolism as a treatment for Huntington's disease. So cholesterol, you can see here, is really linked or thought to be linked to Huntington's disease. So we came to Huntington's disease really from the thinking about cholesterol metabolism and thinking how perhaps cholesterol metabolites could actually be a biomarker for Huntington's disease, which could be used to monitor the progression of Huntington's disease, which of course would be very valuable in any clinical trial. So our hypothesis really was that perhaps a cholesterol metabolite, which is made in the brain and then exported into the bloodstream, maybe that could be a biomarker of the progression of Huntington's disease. So we were sponsored to do a reasonably large study by the CHDI Foundation in the USA. So we looked at 400 plasma samples and 400 CSF samples from controls, from pre-manifest HD. So that's people who've got the genetic predisposition to get HD, and those with manifest HD, once the symptoms started on. And we measured 21 oxysterols and five other sterols. So here's a list of the different metabolites we looked at, and we made our primary hypothesis was based on this one. This 24S hydroxycholesterol. So 24S hydroxycholesterol, it's exclusively made by neurons, and it's only made by neurons in the brain. And 24S hydroxycholesterol, because it's got this extra hydroxyl group, it's able to cross the blood-brain barrier and get out of the brain and into the bloodstream. So our hypothesis really is that if you've got the levels of this 24 hydroxycholesterol that we measure in the bloodstream could be a marker for brain health. Okay, so we did this study and we measured all these different oxysterols. And we measured all the oxysterols. The only one to change was actually 24S hydroxycholesterol. And you see, here's the data: healthy controls in blue, pre-manifest in green, and manifest HD in red. And manifest HD is clearly diminished. So good statistics here. However, a complication actually of this study was almost by definition, pre-manifest patients will be younger than manifest HD. By definition, pre-manifest occurs before manifest HD. So our pre-manifest HD patients were younger than our manifest HD. So we needed to take into account age as a confounder. So we gave all our data to Doug Langman in University of Iowa, and he modeled all the data. So he modeled for males and he modeled for females. But even once you've done the modeling, you can see that manifest HD in red are always lower than the pre-manifest or the controls at all age in the model. And this is also true. This is true in males and also in females, although actually the patterns are somewhat different between males and females. So this data really shows that in the group of patients with manifest HD, the levels of 24-hydroxycholesterol are lower than in the pre-manifest or in the control. So this could be used. This 24-hydroxycholesterol in plasma can be useful in clinical trials as a measure of the effectiveness, really, to see if there's any improvement or a slow down of deterioration. We've also done some other statistics. A colleague of mine, David Skabinski, he's done some binary logistic regression analysis, where he takes basically all the data we have, all the different oxysterol data, age data, also BMI data, and develops an equation or a model which can predict the likelihood of a particular sample falling into the different groups. So he's able to build a model which had 81% accuracy of predicting which group a particular sample would fall into, and it's got an area under the curve of 0.89, which is actually pretty diagnostically good. So where we think that this 24-hydroxycholesterol is probably going to be a good biomarker in HD clinical trials. So what I think is happening is, to be honest, is, as HD progresses, neurons die, you have less neurons available to make the 24-hydroxycholesterol, so you get less of it in the bloodstream. So that's what I think is going on. Okay, so let me just tell you a little bit about what's the focus of a lot of our current work, and this is inborn errors of cholesterol metabolism. So an inborn error of cholesterol metabolism, this will result when there's a defect in one of the enzymes of either the cholesterol synthesis or the cholesterol metabolism pathway to bile acids. So these are rare diseases. So that's the fact that they're rare diseases, they're obviously clinically uncommon. So that means if it's clinically uncommon, it's difficult to diagnose because the odds are the first clinician you see will not be aware of the disease. So it takes a long time to get a diagnosis. However, there are good mass spec assays to actually make a diagnosis. But for each of the different inborn errors of cholesterol metabolism, there's a different assay. So again, the clinician has got to choose which assay to guess. He's got to guess which assay to choose, and he's got to be right. And these assays only performed in specialist labs, and they're only ever done when the clinician's has made a choice of an assay to use. So the result of this is these inborn errors of cholesterol metabolism and biosynthesis, they're really undiagnosed. So, for example, this one, CTX, cerebrotendinous xanthomatosis, which I've got a few slides about this later on. Only 300 cases have ever been reported. However, the prevalence is calculated to be between 1 in 50,000 and 1 in 500,000. So there should be loads more cases reported. So how can the diagnosis of these inborn errors be improved? So one way would be instead of having a different mass spec assay for each of the inborn errors of metabolism, would be to have a catch-all multiplexed assay where you could actually use this assay to diagnose all the different inborn errors using just one using just one assay. And of course, this should be available to the different healthcare systems. So actually, it just so happens that, the method that we've developed based on this enzyme-assisted derivatization, and then this switching into this positive negative ion switching, so we can see the bile acids, really just about covers all the different inborn errors of cholesterol metabolism. So this is a really simplified view of cholesterol metabolism to bile acids. And there are two pathways. There's the acidic pathway at the top and the neutral pathway at the bottom. And all the different enzymes are marked in blue. And if you've got a defect in one of these enzymes, then you'll get one of these inborn errors of metabolism. But the fact that we can measure all the metabolites in these pathways actually means that we can actually identify any of these inborn errors of metabolism. So I will sort of demonstrate that by focusing on this CTX disease, cerebrotendinous xanthomatosis. So as I said, it's 1 in 50,000 to 1 in 150,000. But in fact, as I said, only 300 cases have been reported. So it's autosomal recessive. So there needs to be a mutation in the gene from both your parents. And it results in a defect in the enzyme CYP27A1. So you'll see where that comes in a minute. So if you get this in childhood, you get cholestatic liver disease. So if it presents in childhood, you get cholestatic liver disease. And then there's a good chance of it being picked up because the pediatricians will be aware of CTX. However, if you don't present in childhood, you've got the mutation, but you don't present in childhood, you get to middle age, then you get psychiatric abnormalities and a movement disorder. Then it's incredibly difficult to diagnose because there's so many disorders with psychiatric abnormalities or movement disorders. So it's at middle age, it's really difficult to diagnose. What's also more important is there is a treatment for this for CTX, actually bile acid replacement therapy. So there's a good treatment. So early diagnosis is really key. So if we look at our pathway again, so CTX is the enzyme shown in red. So if you have a defect in CTX, this top pathway, this acidic pathway, is turned off. So all these metabolites disappear. So this first one, 27-hydroxycholesterol, also called 26-hydroxycholesterol, should disappear. So then bile acid metabolism gets forced into this other pathway, the neutral pathway. However, this CYP27A1 appears in the middle of the neutral pathway. So what happens is you get a huge buildup of these early metabolites in the neutral pathway. So actually, you'll see diagnosis can actually be quite simple. So here I show that with a couple of chromatograms. At the bottom, we've got a control chromatogram. In the top, we've got CTX chromatogram. This is from plasma from a patient. So in the control, 27-hydroxycholesterol is decent size, and in the CTX patient is completely disappeared because the enzyme to make it's gone. Instead, metabolism got shifted into this neutral pathway, and these early metabolites of the neutral pathway go up. This one's also a early member of the neutral pathway, which is almost absent in the healthy control. So what we can actually do, if we plot, if we look at the ratio of this early metabolite, which is high in CTX, against 27-hydroxycholesterol, which is absent in CTX, you get a huge value, while in the control, you get a very small value. So the diagnosis by looking at this ratio is trivially easy. So using this method, diagnosis can be done quite easily. And what we're trying to do now in the UK is we're trying to translate this method that we've got from a university into the NHS, the health service labs in the hospitals. But translating a method in theory is very easy, but when you've got a grossly overworked and underfunded NHS, it's very difficult. Okay, so last 5 or 6 minutes, I'll talk to you a little bit about mass spec imaging. So we got into this really when we were doing our studies on mouse brain. Because in the past, when we'd studied mouse brain, we'd taken a whole mouse brain, homogenized it, and look at the oxysterols you'd see in the whole of the mouse brain. But clearly, the levels of oxysterols are going to change in the different regions. So we thought it's really important that we could actually see how the levels of different oxysterols, of cholesterol, of its precursors change according to location. So we got into this mass spectrometry imaging. So let's explain to you what we do. So this is a mouse brain, and we cut sagittal sections, thin sagittal sections from the outside to the inside. And we take a sagittal section, about 15 microns thick, and we just plop it on a microscope slide. So once we've got it on the microscope slide, first thing we do is we spray on, using a pneumatic sprayer, isotope label standards. This allows us to do quantification. Next, we spray on that cholesterol oxidase, that enzyme, to convert those 3 beta hydroxy groups into 3 oxide groups. Then we spray on the Girard reagent. Then we can just incubate and let the derivatization occur. And then we do the mass spectrometry imaging, where we record mass spectra pixel by pixel by pixel. So let's show you some data. So this is the easy bit. This is cholesterol. So cholesterol is really abundant in brain. So there's lots of signals. So this is a sagittal section. And red is high abundance. Blue or black is low abundance. So we've got high abundance in actually these are fiber tracts of the pons and the medulla. We have high abundance in the white matter of the cerebellum, and also in the corpus callosum. Here, this is a ventricle, so we see nothing there. This is a 50 micron pixel size. So to generate this image, it takes overnight basically. So it's quite time consuming, and we can go down to 10 micron pixel size to get more almost single cell resolution. But then as your pixel size goes down, you need more pixels to cover your area. So we just actually do small areas because it's so time consuming. So cholesterol is easy. Now those oxysterols are a lot more difficult, because the problem is they're less abundant. So it's at least 500 times less abundant, maybe 1,000 times less abundant cholesterol. And you got all those isomers. And this image that I showed you, this is a moldy mass spec image, and there's no separation involved. There's no separation of isomers involved. Cholesterol is just one isomer. It's the completely dominant isomer, so we don't need to worry about isomers. With those oxysterols, we've got all those isomers. So when we come to oxysterols, we have to use a different imaging method. So here we change our imaging method to what's called LISA, or liquid extraction for surface analysis. So again, we've cut our brain into thin slices, done our derivatization, and here's the brain on a microscope slide. What we now do, we're now going to do liquid extraction in small areas. Take the liquid extraction and then put it onto an LC system to separate the isomers and do spot analysis. So let's explain to you how this works. So here's our brain again, and we've got, this should be a real time. Let me remove the, where are we? I want to remove the pointer. Let's see, remove the pointer. Okay. So I click on here. This is a real time. So here's our extracting probe. It picks up some solvent. It then goes to the brain, and we have a liquid extraction on a small area of the brain. So it's extracting now, and then any second now it will take the extract and put it into a vial, and the vial will then do chromatographic separation. So we then, this is a diagram of what's going on. We've done that extraction. So it's about 330 microns is the extraction width. We take the extract. Ooh, come along. goes to a trap column. We trap on the sterols and the oxysterols, wash away any excess Girard reagent. Then the trap goes in line with the analytic column. We separate the isomers, and we get the and then we do spot by spot images. So this is for abundances images for 24-hydroxycholesterol, and this is 24-25-epoxycholesterol. So we get high levels of this 24-hydroxycholesterol in the striatum. This is the one that's made in brain and is then exported into the bloodstream. So we get a lot of it made in the striatum, some also in the thalamus, almost nothing in the grey matter of the cerebellum. This one, 24-25 epoxycholesterol, this is about 10 times less abundant, and this is most abundant in the thalamus. So we can do this spot analysis and generate different images for different oxysterols that we see. So what we're doing at the moment really is to actually make a mouse brain atlas, basically, where we've cut the mouse brain in different slices, starting from the outside, going to the center, and we're building this into an atlas. So this is for cholesterol, and then we're doing exactly the same thing for each different oxysterol. So here we've pixelated it into actually millimeter squares, really, to actually speed up the process. So you can see, again, it's most abundant in the striatum. 24-25 epoxy most abundant in the thalamus. We also can do the exactly same thing for the cholesterol precursors as well. So we can see where the different oxysterols are made in specific regions of brain. So this is quite a lot of work, and for each of these pixels, it takes about an hour of mass spec time. So you can imagine when to go through the whole of the brain. It's years of work. So this is a big study. So that's more or less it. So the summary really is that these oxysterol molecules, I think their most important function really is as fine regulators of cholesterol levels. You know, that might be via paracrine mechanisms, or it might be by the cell that makes the oxysterols themselves. Our EDSA method improves sensitivity and adds specificity. Oxysterols can be markers of neurodegeneration. So this is definitely the case in Huntington's disease, but it doesn't seem to be the case in Alzheimer's. So when we've looked at Alzheimer's, we don't really see much difference between the Alzheimer's and the controls. Yeah, that's just the way it is. So we think that 24-hydroxycholesterol can have value in these clinical trials that are being performed on for HD. And I showed you our catch-all method for the inborn errors of cholesterol metabolism. Finally, I showed you the mass spectrometry imaging, so we can get absolute quantification of cholesterol and these oxysterols and cholesterol precursors in specific regions of brain. So we've done this on mouse brain. We've also done it on sections of human brain. We only ever work on a section that can fit on a microscope slide. So we just done it on small sections of human brain. Okay, I think, yes, that's almost it. So this is our group really. So this is Elan, who'd done most of the imaging work. Mosin, who did the HD work. This is Yutin that John mentioned earlier. This is actually a friend of mine, Cedric, who works in Oakland actually. And then two girls which have left us, Manuela and Lauren. And our funders, CHDI, who funded all that Huntington's work. This was the EU actually who helped us out with equipment, and then UKRI. This is the research councils in the UK. They funding this rare disease work. And finally, I thank you for listening. Okay.